Morning, everybody. Thanks for coming out. There's a lot more people here this early than I expected. Really appreciate it. I'm going to get this started, and I'm going to be talking about precision functional mapping, which is something I've been obsessing about lately. Let's see if I can. Sorry. Oh. I clicked it 20 times. I'm a little worried that this is going to run through all my other slides now. This is the least interesting slide. That one isn't that interesting either. Here we go. All right, and I'm going to focus on the approach a little bit. Essentially, I'm going to try to convince you guys that precision functional mapping, and I'll of course talk about a little bit more about what I mean by that, is something you should all be trying. It's doing individual-specific studies, doing deep phenotyping with fMRI. It's very expensive because we get a lot of scans per subject, so it's small n. And the study that we've done that is really available, the Midnight Scan Club data set, we actually only spent $12,000 on 120 scans in 10 people. And then, so this is sort of one end of the spectrum. The other end of the spectrum is the absolutely humongous ABCD data set. It's longitudinal. It's actually a representative sample, which is hard to get with precision functional mapping. It's got 12,000 roughly participants. It was expensive. So I'm going to be talking about precision functional mapping, and then the person who's going to tell you about ABCD is Damien Fair. If any of you don't like the 90s, please close your eyes. That photo is so 90s. He's sitting right there. That's Damien. And he likes to play dress up. So I'm showing some old photos from graduate school to make sure that if you want to talk to him afterwards, you can recognize him. And this is a true story. I don't know if any of you know Steve Peterson, but that's me, Steve Peterson, and Damien in grad school. And Damien has actually been mistaken for Steve Peterson. So real quick, that's Steve Peterson. That's Damien Fair. That's Damien Fair. He's going to tell you about ABCD. I put a black slide in here because these photos of Damien are a little traumatizing, so I need to like center myself, actually talk about some science now. All right, and I'm going to start with talking about a patient of mine. I'm also a child neurologist that really started this obsession with individual-specific precision functional mapping. So I hope this video plays. Let's see. I was just going to talk about this patient. You can just watch the video. So he came into my child neurology clinic for a motor complaint. Essentially was referred by his baseball coach. He's a left-handed pitcher, and you saw him going with his left, and now when he's switching the wrench to his right, you can see how he's going a little slower. So that was the extent of his, and then when he switches again, you can see that he's going really fast. So essentially had this very, very mild asymmetry that only got picked up because he was a serious athlete, and his coach, when he was 14 years old, thought there was maybe something wrong with his elbow. I got a history from the mom and found out that when he was 6 days old, he got really sick and was in the ICU for 2 weeks with low blood pressure, low body temperature, but pulled through, walked at 1 year, talked at 1 year, dragged his legs when he started walking on the right, but then that all got better, so nobody worried about it. So based on that, I thought, okay, he's got probably a perinatal stroke. There's nothing wrong with his elbow. But I thought it would be a tiny stroke given the video kind of shows how mild his deficit is. And the mom didn't really believe me, so she wanted an MRI, and I love MRIs. So we got an MRI. I'm going to show you, this is a clinical scan, so it's not that pretty. And the arrows are for Damien. That's where the problem is, Damien. I'm going to click through some coronal slices, some horizontal ones, but there's just black, there's no brain. There's just CSF there going down, and then these are parasagittal slices. So essentially, much to my surprise of finding a tiny little stroke in the left MCA distribution, I found that he was missing about a quarter of his brain in a symmetric fashion. But he was essentially a normal kid other than the slight deficit with his right hand. He's actually exceptionally good with his left hand. And so I was fascinated. I felt like this one patient was telling me a lot about the brain, about development, about plasticity. There's no way that if this had happened to anybody in this audience at an adult age that we'd still be playing baseball. So I felt like I knew from just one patient there's something special about the newborn brain. And I really like this slide because this is the only use I have for this finger tapping fMRI map that I did as a grad student in Steve Peterson's lab in 2002. But I'm just showing this. It's showing the architecture of what it takes to do left-handed finger tapping, and he's left-handed, so he's great at that. And then his brain, and essentially all the regions that are normally activated are missing in his brain. So there must be something going on. I really wanted to study this case. He'd never had a seizure. He'd never been seen by doctors. Nobody had sort of messed with him. So I thought this would be a really great case to understand some of the principles of brain plasticity. And knowing about patient HM, there's a strong history of learning a lot about the brain from just single individuals. So I got really lucky because I'd seen this patient, and then shortly afterwards, I heard about Russ Poldrak's super awesome myconnectome project where he scanned himself over 100 times. He got the data to people at WashU, Tim Laumann, Steve Peterson, and they figured out how to do what we normally do in groups in single subjects and analyze Russ Poldrak's brain. Here's a plot that I love because they showed that if you have that much data, the red arrows at the 5-hour mark, you can essentially squeeze out all the measurement error. I think they got the R to 0.98, and the remaining variance was due to stuff like whether or not he'd had a coffee in the morning. So you can measure somebody's one true functional connectome. And then up top here is this pretty standard resting state networks or functional networks map. Data from Steve Peterson's lab up top. That's the kind of stuff that we were all used to. And at the bottom is Russ Poldrak's one true functional connectome. And right away you can see there's a lot of similarities. The gestalt is very similar, but there's also some very obvious differences. A lot of smaller pieces, pieces we've never seen before. So the black arrow is just pointing to an example. That's the region of singular Perkin network inside frontoparietal network. And this is Tim Laumann's work, which you can see. What you see here is when you take two seeds that are right next to each other, that in Russ Poldrak's brain, one of them, the green one, is in this little island of singular Perkin network, and then the blue one is right next to it, but in frontoparietal network. And you make two seed 1 and seed 2 seed maps, and you do the difference. At the bottom, you see that in a single individual's brain treated like we did, you get this strong difference map, meaning that they're very clearly assigned to different networks. And up top, with all the smoothed out group average data, other than slight differences in autocorrelation locally, there's no difference. So for those data, it really is true that there's just this giant swath of frontoparietal network. So this was one of my all-time favorite figures. This was really what, after seeing my patient, got us started on precision functional mapping. So we didn't have as much funding as Russ Poldrak, so we started something called the Midnight Scan Club, where I conned my friends into scanning people, and including ourselves, after midnight, because it was 90% off in scanner charges. Here's some of the people. I want to talk about, real briefly, just to give you sort of an overview, about some of their papers. This is Evan Gordon's paper. The description of the data set. We got 4 hours of structurals, 5 hours resting state, 15 hours of task, neuropsych batteries. And then this is what we did. Up top is the group average again, and at the bottom are 10 individual functional connectomes, not based on quite as much data as Russ Poldrak's, but pretty good. I'm number 2. We're still working on what this all means. That's really the hard part with small n's. We did a bunch of validation stuff. Evan did a great job with that. For example, this is validating the resting state functional connectivity boundaries as outlines for hand and face motor cortex. And then the underlay is just a simple HCP motor task activation map and showing how individuals, how perfectly that overlaps. And we did a bunch of other validation stuff that I'm not going to show here. Evan also took our functional network maps and turned them into spring-embedded plots to look at the whole brain as a network. And then here are the 10 brains, sort of zoomed way out like this. And we didn't really expect to find much. We were just kind of, doing discovery science. But what we realized, much to our surprise, was that in these 10, there were 2. Number 2, that's my brain. Number 6, that had significantly lower global efficiency, for example. So 8 out of 10 of the brains had this sort of donut ring structure that makes it easy to get from one part of the graph to the other, any other part. And then I have severely reduced global efficiency. That effect is actually significant by permutation testing, which explains a few things about me. Another one of my all-time favorite papers, Katarina Grattan, wanted to look at how stable is the signal in a variety of conditions across different tasks, across different sessions, across individuals, and across all the people in our sample. And these are actually not real data matrices. These are just the matrices you would expect if you had an effective task, an effective session, an effective individual differences, an effective group. And I'm going to show you the real data now because I want to set this up to convince you that it's mostly about inter-individual differences in resting state functional connectivity. So here's the map, and to the side of it is a bar that quantifies the variance you can explain. So about 50 to 60% is shared across the entire sample, which is not truly representational, so I'll wash you neuroscientists. And then there's another almost 50, another large chunk is inter-individual differences, and then task and session don't really affect the signal much, and actually the effect of task is an interaction with the effect of individuals. So different individuals change their resting state functional connectivity matrix differently depending on what task they're in. And here's sort of a map showing how much of the variance we can explain in the data on the same color scale, showing that there's a lot of shared stuff in the group, which is good news for group studies. There's a ton of inter-individual differences. That's bad news for group studies if you group average. And then task and individualized session really don't explain any of the variance, which is really good news for resting state as a potential clinical tool, because you don't want a marker wildly fluctuating, every 10 minutes. That would not make a good clinical test for neurologists, psychiatrists, and neurosurgeons. And then this is just showing that a fairly simple model, all those factors I talked about, altogether can explain almost all the variance that we see in the Midnight Scan Club resting state data. This is work by Scott Merrick. I'm just giving you sort of an overview of all the cool stuff we've done. Doing the same exact thing that Evan Gordon did in the cortex, in the cerebellum, which is harder, there's worse signal down there. And so here are the same 10 subjects and their resting state functional connectivity maps in the cerebellum. Here these are the data curves for the cerebellum, showing that lots of data, you can get nice replicability in the cerebellum. Not quite as good as in the cortex, but not bad. Scott did a quantification of how much of the real estate of the cerebellum in volume space and cortex on the surface is dedicated to each of the canonical resting state networks. And what's interesting is that the cerebellum is 80% higher order stuff, attention, executive control. And the one network that's grossly overrepresented is the frontoparietal network, which is not something that we expected. It's a very nice paper, again, just looking at the cerebellum in 10 people, where all we did was get lots of good data in just 10 people. Another thing we found in the cerebellum is doing Mark Wakel lab style lag analyses, that the cerebellum is always late with respect to cortex completely systematically, and is not sort of late within itself much, meaning that its timing is all in relation to cortex, not the circuitry within the cerebellum isn't that important, it seems like. So precision functional mapping on 10 people, we found out that the cerebellum is individual specific, just like the cortex, that it really seems to care about whatever the frontoparietal network is doing, and that it's always late in relation to the cortex. These are just some teasers. We've also been doing this on the basal ganglia and thalamus. Having really nice SNR is really helpful there. It's also really nice to have precision functional mapping in smaller structures. So this is just a teaser, but we're doing individual level resting state functional connectivity in the deep gray. This is work by Deanna Green and Scott Merrick. Deanna can't come, but Scott's going to present that. So if you want to know more about that or his cerebellum paper, go to his talk. We're also doing the same thing in the hippocampus, another tiny structure with bad SNR, and we're finding some interesting things. I'm just going to give you a teaser because I don't want to steal people's thunder, but there's going to be a poster about that. Annie Zhang's presenting that from our lab. Chad Sylvester, he doesn't have a poster, so you have to go find him. He's really bald, so you should be able to find him. There's his picture. He's done this work in the amygdala, and the paper will be sent out momentarily. We've also done it in individual child brains. This is a 9-year-old boy. We have 12 times half an hour of resting state. This is the reliability curve of the data. It looks just like the adults. This is for that one subject, his individual functional connectome compared to a small subset of the ABCD. So sort of same finding as in adults. Gestalt's the same, but you get a very specific different pattern in an individual. This is the similarity of that kid against the ABCD cohorts. And then to come back around to my intro patients, we also figured out patient DC's individual functional connectome. And if you want to hear about that, you got to go to Tim Lauman's poster. It's really fascinating stuff. And then the craziest thing we've been doing lately is we're using this to do experiments, kind of like monkey experiments. So I get referred to as monkey number 1 because I casted my dominant arm for 2 weeks, and we scanned every day, 2 weeks before, 2 weeks during, and I think 4 weeks afterwards. Big team on that project, and this is just some of the behavioral data, but we can change the use ratio from usually around 1 to 0.5. You can induce 30 pounds of grip strength that comes back again. And if you want to hear what that does to the brain, go to Dylan Newbold's. He's got a talk and a poster. And then if you're interested in the Midnight Scan Club data set, it's on OpenNeuro. A few people have downloaded. Go check it out. And then here are the people who did all the real work. I'm just taking credit for it. Thanks.